My name is Felix Gremse, and I will talk about quantitative multimodal fluorescence and bioluminescence tomography with the MILabs OICT system. I work in Aachen at my company Gremse IT GmbH, as well as two institutes at the RWTH Aachen University. The goal of our study was to evaluate the MILabs FLT-CT system, which had been upgraded for fluorescence tomography by MILabs in cooperation with EXMI and Gremse IT. We used three different fluorescent probes with IV injection to determine their organ and tumor uptake and measure then afterwards the fluorescence in excised organs. And we wanted to test the multimodal bioluminescence tomography features. Multimodal fluorescence tomography makes use of a laser mounted on the bottom below the mouse, which transilluminates the mouse in a diffuse way, and then many images are acquired with a camera and a filter mounted in front of the camera. And the laser is moving around to cover different parts of the mouse. Then from all this raw data, a 3D fluorescence distribution is reconstructed, which looks a bit like a PET signal. And here you can see some example in the knees and in the spine. Then an organ segmentation is performed, and we get the quantification, the organ biodistribution. The FLT makes use of a special mouse bed, which is compatible with CT, BLT, SPECT, and PET. The mouse is covered between two plexiglass plates, as is shown here. The MILabs system acquires a CT scan automatically before the FLT scan. That data is then used for an automated segmentation of the mouse bed, including the markers which are used for fusion, but also the mouse shape here. From that, an improved optical model is generated. The fluorescence reconstruction makes use of the shape, which has been generated based on the micro-CT data, and this is quite important. So on the left here you can see an example without the shape, and there you see signal in the air and around the urinary bladder. If we use the shape here, it looks much better, and the signal in this example is finally where it should be, in the bladder. The micro-CT data is also used to generate an optical scattering map in an automated way using an organ segmentation. So the tissue types lung, bones, fat, skin, and the remaining parts are automatically segmented and used in combination with fixed coefficients to generate a heterogeneous scattering map. Based on the shape, the scattering map, and the optical raw data, an absorption map is generated. This is important for the optically dark organs such as the heart, liver, and kidney. They are dark because they have a high relative blood volume, which is the dominant absorber in this wavelength. And using such an absorption map is important to avoid a systematic underestimation of the fluorescence in those dark organs. So the three fluorescent probes we used were free dye, Cy7, and there we expected a fast renal clearance because it's a small molecule. We used core cross-linked micelles, they're called CriPec, and they were Cy7 labeled, and we expected a long circulation and tumor uptake. And then we used cationic micelles as a bad example for a nanoparticle, because due to the positive charge, we expect fast liver and spleen uptake and no tumor uptake. As a mouse model, we used the 41 orthotopic breast cancer model. The CriPec micelles which we use are based on a versatile platform. They are assembled by polymers and drug linkers. They assemble to these micelles, can be linked also with active targeting moieties on the surface, but we used Cy7 only in this study. When these micelles reach their target, they eventually will release their cargo and degrade and be eliminated. But in our study we didn't use any drug loading yet. The FLT-CT results from using the Cy7 dye showed that we had a strong liver signal, and then signal in the gut, and then at the last time points signal in the kidneys. The ex vivo organ imaging also confirmed this. It was a bit surprising that we didn't see a strong renal elimination, which would look a bit different. Like this, it would show a strong signal in the urinary bladder. This is an example from the dye Alexa Flow 750. The biodistribution of the CriPec probe showed long blood circulation time here, so you see a lot of signal all over the body throughout the whole time. And then over time the tumor lights up, so you can see that it starts lighting up here. And the ex vivo imaging confirmed these results. So here you can see the tumor and some remaining signal in the liver and the kidneys, which you can see in vivo here as well. The cationic micelles showed a strong uptake very quickly into the liver and the spleen. This remained like that throughout the whole imaging and was also confirmed by the ex vivo data. We didn't see any tumor uptake. After imaging and reconstruction, we used the image analysis software Imalytics Preclinical, which is actually a product of my company, and it is sold with the MILabs OICT system. The software features interactive segmentation operations, is fast because it's GPU accelerated, and versatile. It's easy to learn and has been used for over 100 papers. It's particularly good for multimodal images. The whole body quantification showed some clearance of the free dye after 2 days, down to 20%, whereas the other probes had a longer retention, with the longest retention for the cationic micelles. The liver curve was similar to the whole body curve, and the cationic micelles showed the longest liver retention. The tumor curves showed that only the CriPec probe had a substantial tumor uptake over time. The tumors also grew a little bit over the 2 days, which is normal for these kinds of studies. After the last time point, 48 hours, we had excised the organs and used them for 2D fluorescence imaging. On the left you can see these organs, and then we quantified the mean intensity and compared it with the mean the concentration in the organs determined by the in vivo imaging. And you can see a similar pattern here. For example, the cationic micelles had the highest liver uptake and the highest splenic uptake, and the highest tumor data was in the CriPec probes. And there was some kidney retention here in the free dye and also the CriPec probes. We used all these organ values to generate a correlation between the in vivo concentrations and the ex vivo means. And we got a R of 0.95, and it was significant, showing that FLT-CT can be used for quantitative organ biodistribution assessment. In summary, the FLT-CT shows different organ and tumor curves for different probes. We found a strong correlation between in vivo and ex vivo results, and apparently the tomography resolves quantification problems of 2D imaging. Therefore, we think that it is useful for nanomedicine development. The CriPec-Cy7 probe showed long blood circulation and a strong tumor uptake. We also evaluated the bioluminescence tomography features using a 3 mouse bed. The acquisition takes around 3 to 5 minutes. We used a 3D printed mouse phantom for BLT imaging and inserted a brake light stick into the rectum, a hole in this phantom. The reconstructed image data showed a nice co-localization of the BLI source here in 3D, close to the brake light stick, as we hoped for. The bioluminescence tomography works by acquiring multispectral bioluminescence images using filters. Here you can see an example of a mouse with tumors and metastases, and what happens is that at the higher wavelengths you will see the deeper signal, because the lower wavelengths are much more absorbed by the animal tissue. After fusion and reconstruction, The BLT-CT data can then be used similarly as the FLT-CT data. So you can generate a segmentation here of the tumors and metastases, and then use the colocalization to quantify the amount of bioluminescence in each tumor. In summary, we can say that BLT resolves bioluminescence sources in depth with up to 3 mice and is compatible with micro-CT. Or if you use it with 1 mouse, it's compatible also with FLT and SPECT and PET. So I would like to thank my cooperation partners, both from Crystal Therapeutics and MILabs, and my colleagues from the EXME team, and thank you for your attention.